Hello, and welcome back to the Introduction to BioCyc webinar series, Part 4, Genes, Operons, and the Genome Browser. I'm Alexander Shearer, speaking on behalf of the Bioinformatics Research Group at SRI International, and I'd like to welcome you back to the Introduction to BioCyc series. So far in this series, we've looked at the BioCyc website, what it contains, and how you can access that information. And using the example of arginine acquisition, and arginine biosynthesis, we've looked at compound pages, transport reactions, transport pages, and metabolic pathways. And now we're moving on to genes, operons, and the genome browser. At the end of Part 3, where we were looking at metabolic pathway pages, we decided that we wanted to go to one of the genes involved in arginine biosynthesis, and to do so, we went to the gene on the metabolic pathway page and clicked through. And that brought us to the ARG-B gene page. This is a representative BioCyc gene page, the kind you should find for each gene in a BioCyc database in our Tier 1, 2, and 3 collections. So let's look at a gene page and what it contains from top to bottom. Up at the very top, we have the organism and the name, and down below that we have three buttons for nucleotide sequence advanced, and protein sequence. And these do pretty much what you'd expect. When I click on the nucleotide sequence button, I come to this, the nucleotide sequence for the gene ARG-B. We have the start codon marked out in lowercase green letters here, and then the stop codon marked out in red letters here. And now I'll go back. The advanced nucleotide sequence button takes you to this little dialog box which lets you pick a left and right in position, so you can actually have larger flanking sequences, such as you might want if you were picking locations for PCR primers to PCR out a gene. Then the protein sequence button gives you the translated protein sequence for the gene, assuming it codes for a protein. We can also accommodate genes that code for strictly RNAs, such as rRNA genes, or things that code for small RNAs, etc. Below these three buttons, we have a synonyms line, which gives other names the gene is known by, in this case just an ECK number and a B number, which are E. coli generic numbering systems, but it can also include other synonyms from when a gene was known by one or more different names in different research studies, so that if you're looking at older publications, you can identify the gene even though its name has changed. Below the synonym line, you'll find these two forms of classification. The first is multi-fun, a classification system that we've been using for E. coli genes for quite a while. After this, you'll find Go terms, which we began supporting in 2006. They're just the same Go terms you'd expect to find anywhere else. If you click through on any one of these terms, it will take you to the definition of the term, and it will show you its parts, part of, parent classes, and so on. Now we'll go back. We initially used an automated conversion for BioCyc from multi-fun to Go, and we are currently in the process of making the Go terms in BioCyc more specific. And should you decide to adopt your own database and run it yourself, and you're curating it, you can of course curate whatever Go terms you want, maybe Go terms to match the genome annotation you're working off of. Below this is this line map position, which shows the map position in nucleotides and senesomes. And you'll also notice it's an active link that will go to the genome browser, which we're going to come back to and talk about a little bit later in this presentation. Below this is the sequence length and the calculated number of amino acids, and then a link to products of the gene and any protein complexes or other complexes they may be a part of. In this case, we see that the product is ARG-B and that it's a subunit of the homomultimer acetylglutamate kinase. Then we have reactions catalyzed by the enzyme, pathways that's involved in. The gene reaction schematic, it's another thing I'm going to come back to later in this presentation, but very briefly shows the connection between genes, proteins, and their reactions. And now as I page down, you'll see a genetic regulation schematic, much like the one you saw on the pathway page, but constrained a little bit because we're only looking at genetic regulatory units that contain the gene whose page we're on, in this case ARG-B. And here we can see that ARG-B is co-regulated with ARG-C and ARG-H, which is not surprising because they're part of the same operon, and they're all suppressed by ARG-R. And as we go over this, we see that that's the ARG-R transcriptional dual regulator. Under this, we have what we call unification links, as I talked about a little bit before. Unification links are links to other databases for the object in question. So for example, here we have links to the coli strain collection CGSC and to the expression dataset collection OUMCF. And so you can click through here and go to your gene of interest. If you're making your own database using pathway tools, you can link out in a systematic fashion to anything that follows a systematic labeling method for its items. So UniProt for proteins, an appropriate gene database for your genes, etc. Below this are ortholog links to orthologs for this gene, ARGB, in related organisms, and then this button for the Align and Multi-Genome Browser. I won't be showing the Genome Browser in this webinar. It will be discussed in the Omics and Comparative Genomics webinar series. The Multi-Genome Browser lets you compare a gene with its orthologs across the entire BioCyc collection. It's really useful, and you'll want to go and look at the Omics and Comparative Genomics webinar to learn how to use it. Below this, we have the gene in its local context, as we helpfully say here, not to scale. Again, we have the little question mark to explain what's going on, but basically what you're looking at here is the gene. There it is, ARGB. You notice when I mouse over it here, I get the gene name, I get its location in nucleotides on the genome, and I get its product, the protein ARGB. And you get everything else that's co-regulated with it, ARGH and ARGC in its operon here. And then one gene on either direction, ARGE and OxyS. Below this, you'll find transcription units involving the gene of interest, or adjacent to the gene of interest in this case. And what you'll see here is that ARGB is part of this ARGCBH operon, that they're regulated by ARGR, and here's the promoter, ARGC promoter. Now notably, since I have a tooltip showing up, this tells me that this is clickable. And so clicking on this will actually take me over to what we call the transcription unit page for this transcription unit. So I'm going to click on this now. And I find myself over here on what we call an E. coli transcription unit page. So this is typical for BioCyc transcription units in a Tier 3 database. These are going to be predicted for all of our bacterial databases or microbial databases, pretty much anything non-eukaryotic. And we call it a transcription unit page rather than an operon page because not every one of these is strictly an operon. Many of them are single gene transcription units. In this case, we are looking at an operon, ARGCBH, and a transcription unit page has a number of features. Here we have the promoter, we have regulatory proteins that bind near that promoter and that regulate its activity, and we have the genes regulated by that promoter. We have a dashed line in this case to indicate that the evidence for the extent of this transcription unit is not considered high quality. And there's a little bit more discussion elsewhere of what that means, but there is experimental evidence for it, but we've decided that we can't say for sure that maybe it doesn't go farther than that, for example. On a transcription unit page, you'll have details about the promoter, citations that support the existence of that promoter. If we know which RNA polymerase sigma factor it uses, we'll have that indicated here. If we have the absolute position of the transcription start site and the position relative to the start of the first gene. And you'll notice here that when I say promoter, I'm really pretty much talking about the conceptual presence of a promoter, but the specific details go to the transcription start site, because of course the actual concept of a promoter, the -10, -35, etc., is a little fuzzy. And now if I page down a little bit, we do also show, when we have the experimental evidence for it, binding sites for transcription factors that regulate that promoter. And again here you see evidence codes, and this has a lot of evidence. We have evidence from binding of purified proteins to the promoter, we have evidence from site mutation, and evidence from cellular extracts in this case. And if I click on this, of course, it will give me all the details about what all of those mean. We also find out that this site regulates some other transcription units, which is why they showed up on the gene page for ARGB. And in addition, we can expect to see the position of this binding site relative to the transcription start site, and that's the center of this binding site, and its binding area, its footprint here in capital letters. And then of course the ubiquitous references at the bottom of the page, letting you know where all of this evidence came from. And of course if you're looking at a Tier 3 database, these will all be predictions, and you'll actually see computers, those little computer icons on the side here or down by the promoter to indicate that you're looking at a predicted transcription unit.